Hi, my name is Kristen Mann, and I am completing the Module 4 application activity. So, the steroid hormone that I chose was cortisol, and this is the pathway. So, cortisol is ultimately released by the adrenal cortex, but this is due to the hypothalamic pituitary adrenal axis. So we're going to start with the hypothalamus. It secretes corticotropin releasing hormone, CRH, and that in turn stimulates the pituitary gland to secrete ACTH, which is the adrenocorticotropic hormone. And this hormone travels through the bloodstream and binds to the adrenal cortex. So cortisol is created in the zona fasciculata of the adrenal cortex, and that is also where it is released and is released into the bloodstream. So let's go back and talk about how cortisol is made. I already said that it is created in the zona fasciculata of the adrenal cortex. Within the cells of the zona fasciculata, ribosomes make the proteins and send it off to the rough and the smooth ER to be altered. And after all of this modification, the proteins are moved via transport vesicles to the Golgi apparatus, and that's when the Golgi apparatus processes the vesicles, and then it can send them out by secretory vesicles. And it travels through cell membranes to glucocorticoid receptors. And these receptors can be found on the brain in places like the hypothalamus or the pituitary gland. It can bind there and create some negative feedback, so it won't be produced as much. But it can also be found on the liver, the lungs, adipose tissue, and this is how cortisol works to regulate our body's stress response. So the protein hormone that I chose was glucagon. Glucagon is produced in the alpha cells of the pancreatic islets. So the goal of this hormone is to make sure that the blood sugar does not get too low. So low glucose levels trigger the alpha cells to secrete glucagon by exocytosis. So when talking about the path through the endomembrane system, cortisol and glucagon have similar paths. So this secretory protein is made in the ER as well, and is moved to the Golgi by transport vesicles where it's manufactured. And then, like I said, it exits the cell by exocytosis. And this is secretory vesicles. It makes a secretory vesicle and then goes through the plasma membrane through exocytosis. And once this glucagon travels through the bloodstream, bind to 7 transmembrane receptors, which are just the glucagon protein receptors, and these are on the plasma membranes of mostly livers and kidneys, but can also be found on the heart, the spleen, adrenal glands, and other tissues. The binding of glucagon on these receptors triggers the G protein, which is on the other side, the intracellular side of these receptors, and it triggers the G protein to activate adenocyclase. And this adenocyclase then creates CAMP, which is cyclic adenosine monophosphate. And this CAMP activates the protein kinase A, or PKA, which activates CREB, the CAMP response element binding protein. And this aids in gene expression and allows glycogen to be broken down and moved to the bloodstream. So last but not least, we have the intracellular proteins, and here specifically we're talking about respiratory enzymes, and these differ from the other two in that they do not leave the cytoplasm, they do not leave the cell, so they go through the transcription. So DNA is used as the template to make RNA in the nucleus, and then it goes to the ribosome where it makes the proteins by translation. And then it does not go to the smooth ER, the rough ER, or the Golgi apparatus. This is because these proteins are not membrane bound. This concludes my presentation. Thank you.